now. Um, I wanted to say a couple of things, one of which was I am very sorry for falling behind on my grading and posting our PowerPoints. Today's was awful. I really apologize. I will get those. I'm going to get ahead of those. I'm going to get caught up on that so that you guys have, the groundwork that you can work ahead if you want and have them at very least to review before our class. One of your classmates emailed me yesterday and they're like, "Hey, would you mind posting them?" And I was like, "Absolutely." I can get them up on Sundays before. And this was like Sunday afternoon, and I promptly got distracted with other stuff. And I'm sorry about that. But I will get caught up, and so you guys have that. Um, and I'll get caught up so that you guys don't have to, you know, email me requesting something that's kind of a low-hanging fruit for the remainder of the semester. With that, um, I have the practice exam up. You guys do not have your exam until the 19th. So that means we've got this week of class, next week of class, and then on Monday the 17th, we're going to review. Um, and so you can look over the practice exam, obviously. Um, and anything that comes to mind, obviously ask me between now and then. Um, but if there's something that surfaces, the 10th, the 17th is going to be a really good day to kind of clear the air on everything. Um, I feel pretty good about the depth and the breadth of the practice exam that I've got posted. Um, I feel like that's going to be a pretty good extension of what we'll be seeing between, well, this week and next week. Um, and then, I will just continue having class. If anyone has office hours that they want to meet with me for, great. I'm a true believer in office hours being like a complete amnesty period. And what I mean by that is if you join my office hours and you say, "Hey, Dr. Gray, or hey, Joel, can we meet in a breakout room?" And I'm like, "Sure." And you say, "What's carbon?" That's fine. That's fine. I want to meet you where you are, and I want to help you kind of, clear up anything. So we have our two activities that are posted. We've got our three quizzes that are posted. I added notes to those quizzes of, hey, most of this stuff from, I think it's A10 comes from last Wednesday's material. A11 is mostly reflective of today's material. And then A12 will be stuff on Wednesday. So just to kind of keep that in mind, you have two attempts on them and everything like that. And one other thing, in the event that you have accommodations and I overlook the time, I'm really sorry about that. Unfortunately, I can't duplicate the quiz, but every time I make a quiz, I try to go back. I've got a note of whose names that I need to add the additional time for. In the event that you see, hey, my additional time's not on there, please send me an email. I'll take care of it as soon as I can. Um, with that in mind, I'm going to go ahead and jump over to, do you mind saying where the practice exam? Yeah, sure. So I got let's see, the practice exam on our Canvas page. I just got a private message asking for that. I believe that if you just go to quizzes on Canvas, it would be on the bottom there, but let me go ahead and post, let's see. I'm going to edit my Canvas page and I'll just post it on, let's see. Let's see. I'm going to post it that week of, I'll say practice exam. I'm posting it as a link in the column next to day 10. Um, and just so everybody knows, that practice exam should be categorized as, and I believe it is categorized as not being a part of your grade calculation. So you can take that. Okay, great. You can take that quiz or that practice exam a solid 999 times, get the feedback, and I believe the conditions are set up so it shows you your your score. Did you say whether or not we were going to make Yes, previous quiz answers. I I I guess I've done it for my other two classes, but I forgot to do it for you guys. I will do that tonight. I'm going to get all those old quiz answers available to you. Another thing that I'm going to do probably tonight is export all of the Zoom questions, Zoom poll questions, and let you guys see the correct answers for those. Um, and then I'm also going to work on the discussion questions because those are all, pretty darn good multiple choice questions that you guys have been authoring. I'm going to compile those, and kind of cull through those. Like, here's a good one, here's a good one. So you guys have another resource and a peer developed resource. So I want you guys to look at those. And I'll see about putting them as a quiz on Canvas, because then it's something that you guys can, you know, answer and everything. At any rate, those are some of the things that I'm going to work on this evening after class. Is there a key to any of? Not yet. I will take care of that. I have those. I just need to post them. How many questions will the exam have? The practice exam is just like your actual exam. Um, Sorry, let me write this down. Assignment keys. Um, your practice exam is 40 questions, 35 of them multiple choice, five of them multiple select or multiple answers. You should expect your actual exams to kind of mirror that. And so, you know how long we'll have for the first test? Your test is all of your tests. So each of your in-class exams, because our actual class is for a total of 80 minutes, you will have 80 minutes for that exam. Um, and then your final exam is the only thing where you'll have, I think it's 2 hours and 30 minutes. So yeah, 150 minutes. Yes, he did say five for no, he didn't say five for your response. He said five multiple answer. Can you repeat what? The exam will consist of approximately 35 multiple choice questions and five multiple answer questions. So that would be a question that's like, select all of the true statements. Or, a good question for that would be, a list of 10 different amino acids, and you have to select all the polar amino acid residue or polar amino acids. Um, and I saw at least one of your classmates raise their eyebrows, indicating, I too think that is a good question. So if I lose internet connection, um, that's yes, in the event that something goes bonkers with your computer or your internet, yes, you can re-enter. Your time does not stop though. And that's not something that I'm a regulator of or anything like that. Or I don't believe that I'm a regulator of that. If I am, I don't know how I am. And I apologize for that. So, enzymes. Um, could anyone add to the chat like any short term or thought that comes to mind whenever you hear the word enzymes? Catalyst. Perfect. I love it. Now, I think that's a perfect way to think about it. And one kind of, oh, proteins that make reactions go faster. Absolutely. If I've said this before, where are the Zoom recordings? The Zoom recordings are on our homepage. When you go to our Canvas page in a web browser, the page is divided up into a number of different columns, and you'll find recordings as one of those columns, and just scroll down and you'll find day 5, day 6, and so on. I link them every day, or try to do them on the same day. And then one of your classmates added ASE. Yeah, because that suffix is very commonly associated with an enzyme. Now, with that said, I think the heart of this all that is worth remembering is when you talk about an enzyme, don't lose sight of the fact that you have a chemical reaction taking place. And one example would be something like if anyone's ever heard of the enzyme hexokinase. Hexokinase is one of the first enzymes in sugar metabolism. Now, what hexokinase does is it takes the sugar, glucose, most commonly glucose, and ATP, and I'll just label that as a high energy compound, takes those two things and makes glucose 6 phosphate and ADP. Okay. Um, an enzyme is going to do that. Now, with respect to that, think about this as like a third party that basically makes a reaction happen and facilitates that reaction to happen. I will do that. Um, it facilitates a reaction to take place. Now, one of the questions that I'd like to ask you guys as a poll question related to enzymes is coming in just a second. And this is just kind of maybe you've heard this kind of anecdotally. Um, I gotta find the exact question that I'm looking for. Is it three? No. Four. Yeah, here we go. Enzymes are made of amino acids most of the time, though ribozymes have enzyme activity. Sugars, I don't know, anger, or amino acids, always. The answers are kind of rolling in the direction that I was kind of anticipating. The answer that is rising to the top is the first one. Amino acids most of the time, though ribozymes have enzyme activity. And that's absolutely correct. I'm going to go ahead and end the poll right here. For quite some time, it was believed that amino acids were the sole building blocks of enzymes. But then it was revealed, and evolutionary biologists and biochemists kind of worked together on this one, and they concluded that, hey, RNA molecules, when they fold in a specific way, they can catalyze reactions. And that's a really significant thing because when you think about hexokinase, one of the things that I'd like you to think is, well, this is a protein and it's made up of amino acids. And when you think about catalyzing a reaction, or a chemical reaction taking place, I mean, this sort of definition goes back to like Gen Chem 1. What is a chemical reaction? You're making and breaking bonds. Okay. So as you're making and breaking bonds, and you think about a protein doing that, and you know, think about our different amino acids and think about the different classes of amino acids, the types of interactions that amino acids can have. If you think about something like arginine, arginine's a positively charged amino acid. If you think about, let's go with GLN, glutamine. Glutamine is really good about hydrogen bonding. These sorts of interactions, these different, I'm going to say R groups, are what drive the ultimately the making and breaking of bonds. Now, with that in mind, for the time being, we're going to focus and really kind of appreciate enzymes as being an example of a protein. Um, yes, a ribozyme is a RNA molecule that does have catalytic activity, but if you think about the amino acids that make up a protein or the amino acids that make up an enzyme, I think it's a little bit easier to visualize those as different R groups and ionic interactions that can take place between them. Now, to kind of drive that point home, here are some examples of enzymes and their reaction rates, both whenever an enzyme catalyzes a reaction and when an enzyme does not catalyze a reaction. Now, some of these terms we're going to get into, um, but what I want you to look at specifically is this last one, our rate enhancement. This is basically a way of kind of quantifying how much does an enzyme speed up a reaction. And if you look at this first enzyme, OMP decarboxylase, this is one of the fun things about enzymes. Um, does anyone have an idea of what type of reaction or what sort of or what does an enzyme named OMP decarboxylase do? And what I would recommend for thinking about that is I'd start at the end of decarboxylase and go from there. What do you think that it does? Got to vote for a kinase, breaks down carbox. It removes a carboxyl group, decarboxylase. This is one of the instances where biologists and biochemists can kind of a little bit they give a little bit of a explanation as to what that enzyme does. Now, decarboxylase. Well, this is a basically an enzyme that removes CO2 from a larger molecule. And OMP, just if anyone's playing along at home, that is ornithine monophosphate, if I remember correctly. Ornithine is a amino acid, but not a proteinogenic amino acid. Now, that's just, yeah, that's fun to know. And it's also fun to know that ornithine, does anyone want to guess where it was first discovered? What organism? Birds. Absolutely. Because the study of birds is ornithology. That's one of my top 300 favorite words. Okay, so what an enzyme is going to do is it's going to speed up a reaction tremendously. If you look at this, 1.4 times 10 to the 17th. So the enzyme catalyzed reaction, well done, well done. 1.4 times 10 to the 17th is how much faster the enzyme catalyzed reaction is than the uncatalyzed reaction. So that's like the tortoise versus the hare. The tortoise is going 1 mile an hour. The hare is going 1.4 times 10 to the 17th miles per hour, considerably faster. And I would love to say what that number actually is, but I think it's like octillion. It's not octillion. It's like hexillion or something like that. So enzymes speed up reactions tremendously. Now, whenever we think about and talk about enzyme catalyzed reactions, there's a couple of things that are worth noting. So this image over here on the right, a little bit to the right, is a good one to look at. And anytime you see something like this, I want to walk through this slowly because I remember seeing an image like this and being told, yeah, this is an enzyme catalyzed reaction. And my response was, that is, ooh, that is something else. How about that? Wonderful. Anytime you see a model like this and you're like, I don't know what the heck I'm looking at. First of all, be patient with yourself and work through it kind of deliberately and find what are the things that stand out to you. Is there anything in this figure that stands out that maybe we've talked about recently? Got some nods of yes, and you're probably seeing our alpha helices and beta sheets. Absolutely right. That's the sort of thing that I'm going to look for and be like, okay, my protein, my my polypeptide is highlighted in kind of a gold amber color. Okay, that's worthwhile for me. So that right there is my enzyme. Now, what about all the nonsense in the middle? Okay, I've got some red atoms that are, chances are those are probably carbons, the yellow ones, and there is an IUPAC color scheme, and I think that yellow is typically sulfur. Um, But that's sort of what I'm seeing with respect to those molecules in the center are this. Let me go ahead. That thing in the middle there, that's my substrate. My substrate is what an enzyme is going to act on. So when you look at that thing in the middle, those are your substrate or your substrates. Now, it's worthwhile to kind of, anytime you see a model like this or an enzyme mechanism, to use those as your kind of beacons and say, okay, that's what I can recognize. Now, within that, one of the things that's a little bit more subtle to recognize is when you see this substrate, notice how this one has bonds connecting all of those atoms, but then there's a little bit of a space between that and this one right here. It's very subtle, but this red atom and this blue atom are not connected. That is likely the bond that is going to be formed between these two molecules. So that happens to be the active site of our enzyme. Our active site is where basically the action or the chemistry, or if you want, you can call it the magic, is going to happen. Now, this is a good example of this is called a ribbon diagram. Whenever you visualize a protein, there's a couple of different ways to visualize them. One great thing about protein structures is if you go to pdb.org, well, PDB stands for Protein Data Bank. And so anyone who's ever resolved a protein structure, whether it's by NMR or X-ray crystallography, typically they're going to deposit that structure at the PDB. Now, the most common ways that you visualize a protein structure is in this sort of ribbon diagram here, or this one right here, a cloud or a space filling model. Okay. They're showing different things, and these different models, they're kind of personal preference, which one do you like more? But also they show a little bit more of well, the space that's filled by an enzyme or a protein or a molecule. Now, with this one in particular, we've got a couple of different highlighted regions. That black molecule, that skeletal structure, there's our substrate. Now, we are not looking at the same enzyme as we were previously. Okay, so we've got our enzyme, which is this cloudy white thing, with also the red and blue regions highlighted. Now, the blue region is the specific amino acids that are involved in binding to our substrate. I do not expect you to look at this and be like, oh, well, that's obviously an arginine, there's a lysine, there's a leucine, or anything like that. This is just space filling, so I don't want you to get distracted or nervous about that. Now, we have a substrate binding site, which is the part of the enzyme that's going to bind to our substrate. Then we've got adjacent to that substrate binding site is our active site, where our actual reaction is going to take place. Now, does anyone have any questions at this point in time about enzymes, substrates, binding sites, any of these models that we've looked at? Okay, so these structures that I've shown you so far, I just want you to have context so that in the event that you see them in the future, you kind of have a reference point. This is not like processing a protein structure is not a part of this class. That's an entire field of structural biology and understanding structures. The great thing about this and structural biology is it shows a good approximation as to what does the 3D or tertiary structure of a protein look like when it's interacting with its substrate. And what are the, in addition to that, with some of these models, you can say, okay, well, here's my substrate. This is a, I shouldn't have, yeah. This is a lysine right here. This is an arginine right here. Hmm. I wonder if those amino acids are involved in breaking a bond. Now, enzymatic reactions, they're going to follow a little bit of a pattern. So here what we have is an enzyme and a pair of substrates. Notice how I said a pair of substrates. Um, are there enzymes that they have only one substrate, one molecule that they work on? Yes. Are there enzymes that have two substrates that they work on? Yes. Are there enzymes that have three or four? Yes, yes. Here's our enzyme. This enzyme in particular is going to have binding sites for two different substrates. We'll call them substrate A and substrate B. Now, substrate A and substrate B will bind to our free enzyme. Boom. Enzyme, substrate A, substrate B. Now, when substrate A and substrate B are in contact with the enzyme, they're closer together. A and B are closer to one another. So if you think back to like, you know, your study of kinetics in Gen Chem 2, you talk about collisions. If you can increase the number of collisions, what does that do to the rate of your reaction? Makes it go up. Speeds up your reaction. Is that an absolute? Not really. It's pretty darn likely. If you can increase the collisions, yeah, more than likely you're going to increase the rate. But those two molecules have to be in the right orientation because, well, you have to increase those collisions, but you have to increase the right collisions. Now, an enzyme, what that's going to do is it's going to grab onto your two substrates and put them kind of perfectly oriented with one another so that their collision can take place. It's going to facilitate that collision. This right here, when your enzyme is bound to your two substrates, we use something known as the Cleland notation. And that Cleland notation is the ES complex. So does anyone want to guess what the E stands for? Yep, enzyme. Don't worry, I heard you guys all say enzyme through the Zoom. So that's perfect. S stands for substrate. This in particular is an example of a bi-substrate reaction because we have two substrates. Now, when those two substrates are basically interacting with the enzyme and they're in close proximity to one another, well, we can have a reaction between substrates A and B. And we denote this with ES star. So that's kind of like reactions taking place. So ES means that the enzyme is bound to your substrate or substrates, and then ES star indicates our reactions taking place. Now, after that, oopsies. What happens? Well, when A and B are in contact with our enzyme and they're in contact with one another, well, both A and B can change. A can become C. B can become D. Now, the way that I think is helpful to visualize this is these characters, A, B, C, D, that is simply, they're variables, they're placeholders. But when you do lots of kind of descriptive modelings with letters, what I think is helpful to do is just to imagine, let's say that A is CH3COOH, acetic acid. And let's say that B is NH4. Is everyone with me there? This is B. Okay. Now, let's imagine part of reactant A is transferred to reactant B, and part of B is transferred to reactant A. What we would get then is this. Oh, let me rewrite it. NH4 plus D becomes NH3 COOH. And A becomes C or CH4. Does everyone follow me there? Okay. Part of molecule A moved on to B, and what we're left behind with is methane. We've got acetic acid becomes methane, ammonium becomes this ammonium with a carboxylic acid. I don't know the name of that. Probably should have picked a better example. Can I repeat the process? Absolutely. Let's go back for just a second. And let's say that substrate A is this guy, CH3 COOH. B is NH4 plus. These two substrates come in contact with our enzyme, and our enzyme says, fantastic. Cleveland notation is that ES. So it seems silly that we have a specialized term, but Cleveland notation is E for enzyme, S for substrate, and P for product. I got to be honest, I'm a little bit jealous of whoever Cleveland was because they got their name associated with letters. And, you know, big ups to them. Okay, so we've got CH3C, let me rewrite, sorry, COOH is substrate A, substrate B is ammonium. They come in contact with our enzyme. The atoms of those two molecules can be shuffled around. And what we get is, well, the CH or the COOH from acetic acid gets transferred to NH4. And then one of those hydrogens gets transferred to CH3. So we end up with CH4, which is C, and then NH3COOH, which is D. The atoms of these two molecules just get shuffled around. That's essentially what this process is. So this forms, well, our enzyme is still bound to our two products. Here, this EP stands for our using that Cleveland notation, our enzyme product complex. So we've got some important complexes there, and those are EP, which is the enzyme product complex. The products have not been released quite yet. And then ES is the enzyme in the substrate. Now, with those are going to be released, our free enzyme is present, so it can start that all over again. Great question. NH3 still has a charge because what we're actually looking at, let me redraw it. You know, this is just kind of a hypothetical molecule, but COOH H plus. That's what we're looking at. So that nitrogen still has 4 bonds going to it. And just so you know, this is just like a filler. These are just, what I'm trying to show is that part of molecule A, right here, gets transferred to molecule B. So that's all I'm looking at there. Okay, now those are released. We're good. When they are released, our Cleveland notation is E plus P. The enzyme and the free enzymes and the free products are both there. So a way of looking at that, and this notation is something that is going to be important for when we continue our conversations about how enzymes are inhibited, how their function is inhibited. E, when you just see E by itself, that indicates the free enzyme. That enzyme is not bound to anything. It's floating around. It can pick up the substrate and catalyze the reaction. Free S, or S alone, is the free substrate. So that substrate is not bound to the enzyme, and that's something that we can kind of manipulate. ES means that we have a complex. Has the reaction actually complex? The reaction has not actually taken place yet. ES star, or asterisk, means that a reaction is kind of actively taking place. And it's going to take place very quickly, but that star denotes the reactions taking place. EP indicates that our product has been formed. The product has not yet been released from the enzyme's kind of grasp. And then E plus P indicates that we have the free enzyme and the free product. Now, the great thing about a biological catalyst or an enzyme is what can you tell me and add to the chat? What can this E do? It can do the reaction over again, over and over and over again. It can continue doing this. Okay, now all of this is a good foundation for understanding an enzyme. Now, I want to ask you this poll question right here. All enzymes have only one substrate. True or false? This is going in the direction that I kind of anticipated. Now, don't get mixed up because when you see that E plus S, that kind of makes you think, okay, a singular substrate. Well, I can give another few seconds, but we're looking at 92, 93% of the class saying false. Now, do I mean at a time? No, because an enzyme could bind two different substrates. It could bind one substrate. Kind of depends. So I'm going to go ahead and end my poll, share the results, 91.9, and now we're going to go back. Now, understanding that E and S, that's going to be valuable, like I said, for understanding mechanisms, and it's also going to be valuable for understanding inhibition. Now, when we talk about, go ahead and just give me an emoji feeling, like an emotion. Oh, that's what, just put that one together. Give me an emoji in the chat of your feelings when I say the word mechanism. Yeah, yeah. Okay. Upset. Yeah. Fair enough. Respect. Okay. Yep. Green throw up. Uh-oh. Cozy. Oh, lucky. Okay. Um, cheeseburger. I like it. Um, enzyme mechanisms. If I think back to organic 2, I think organic 2 is pushing electrons. It's an arrow to go to there and there and there. This is our nucleophile. This is our electrophile. We will use some of that terminology. In the event that that's something that you need a little bit of a refresher on, I will highlight this is an electrophile. This is why it's an electrophile any and every time that we talk about them. I want you to feel comfortable and confident whenever you hear those terms. And so just be patient as we work through them. Now, when it comes to enzyme mechanisms, there's a couple of different things at play. And one of those is, well, we have to think about, and then we also think about the mechanics of an enzyme mechanism. So an enzyme has to bind to a substrate, so it's going to be flexible. Enzymes are also going to be kind of specific. There's a couple of different components of an enzyme that are going to be influencing how that reaction takes place. So there are some instances where a reaction is going to take place with the help of a coenzyme. Now, when it comes to binding specificity or substrate binding, there's kind of, I guess, not necessarily two theories, but there's like an original theory of enzyme substrate interactions was that this little lima bean shaped thing right here is our enzyme. That's made up of amino acids. That has a 3D shape that has like a perfect complementary region for a substrate. There is an idea that, yep, that's what happens. The enzyme has a site or a shape that binds a substrate perfectly, and that's all there is to it. And whenever those that active site, that substrate binding site matches those perfectly. Whenever those two things interact and it's a good match, great. This was referred to as the Fischer mechanism. Now, a couple of years later, there was kind of a new idea that, well, an enzyme and proteins are kind of flexible. They've got mobile regions to them. And so I believe that it was Daniel Koshland, I can't remember his first name or Daniel or Roger Koshland, who came up with this mechanism that is commonly known as the induced fit model. And as is the case with most theories, for the longest time, the Fischer mechanism, also known as the lock and key model, this was kind of the emerging idea. This was like, yeah, this is how enzymes and substrates interact. There's a lock and a key that fits perfectly, done. Then people were noticing, well, the structure of this substrate doesn't really match with what I think the structure of this active site is. So, maybe what happens is that both the enzyme and the substrate have to give a little bit. And so the idea is that, well, this shape, if you notice, I like to look at different parts of this cartoon active site and be like, well, this region right here kind of matches up with that, and this kind of matches up with that, and so on. Well, these kind of complementary regions, well, what happens is that substrate comes in and the enzyme closes in on it so that it induces a change in the shape of the protein. So there's the two major theories were the induced fit model and the lock and key model. Is one right and the other wrong? No. Some of these are great explanations for some enzymes, and some of these are great explanations for other enzymes and how they work. Now, when we think about an enzyme and the active site, well, there are some instances where that active site kind of closes in and more or less clamps down on a substrate to induce that fit and induce that shape change. So what I think is good to think about is with respect to an enzyme is I kind of think about it as like a baseball glove, and you could be playing catch with a ping pong ball or a baseball. There's a general shape that complements, and that baseball glove can close in on it. If you're not a baseball person, that's okay. I like to think that I am, but I know for a fact that I am horrible at baseball. But if I tried out today, I'd probably make every team at the professional rank in not the United States. So, when we think about an enzyme, though, and we've talked about rates of reactions and how they expedite rates of reactions, it's important to look at a free energy diagram. So here what we have on our y-axis, and maybe it's been a minute since you've seen one of these, your y-axis is free energy. Your x-axis is the reaction progress. On the far left-hand side for our reaction progress, this would be our start. And then the far right would be where our reaction concludes. Now, what this is showing, this is what we pretty much always show, is we have a reaction that goes from a high energy to a low energy. This is overall going to be kind of a favorable reaction. Now, what you have to do in order to catalyze this reaction is you have to increase, or there has to be basically an influx of energy in order for this reaction or these reactants to go from our substrate to our product. So from high energy to low energy, we have to basically ride to the top of the roller coaster in order to ride down. Now, what an enzyme is going to do in this case, so this is our uncatalyzed reaction. We've got this uncatalyzed transition state. What an enzyme is going to do is not alter the free energy difference between the substrates and the products, but instead what an enzyme is going to do is this right here. And I see a question in the chat. I will get to it in just a second. What an enzyme is going to do is it's going to reduce the activation energy. Basically, say that that roller coaster doesn't have to climb as high in order to go down. Now, the question in the chat, did you say all in? No, there are some enzymes that kind of follow that induced fit, and then others that follow the lock and key. So it's kind of these are two emerging theories and two valid theories for different enzymes. So one of the things that's important about an enzyme catalyzed reaction is, well, our transition state free energy is reduced. So what does that do for the forward reaction and the reverse reaction? It actually kind of helps both of them. Going from the substrates to the products, well, it reduces our activation energy. So there has to be less energy input. Then to go from our products to our substrates, well, our transition state for our products is a heck of a lot higher in an uncatalyzed reaction than it is for our substrate to our products. But then it's just a little bit lower if you're going in the reverse reaction or in the reverse direction. Sorry. So an enzyme is going to enable, facilitate, help out both the forward and the reverse reactions. So if you're thinking, well, what does an enzyme do? It doesn't change the overall free energy. So this detail right here, no overall free energy change, because the overall free energy change of a reaction is the energy difference between the free energy of the substrates and the free energy of the products. What an enzyme is going to do is it reduces the activation energy. So the enzymes reduce activation, ACTIV, free energy. Okay, so essentially what this is showing is, or what I want to convey here is substrates A and B. If we go back, what that enzyme did by kind of capturing them was position them properly, bring them in close proximity to one another so that they can kind of collide in the perfect orientation so that the reaction between them can take place more easily. In the absence of that enzyme, what do you have? Well, you have to wait on that collision to take place, and you have to wait on that specific collision to take place. So this is kind of explaining the energetics of an enzyme catalyzed reaction. Now, what enzymes are going to do is they are going to lower the activation energy. They are going to catalyze reversible reactions as well. They are not going to change your overall energy, and they are not going to do anything to the equilibrium concentration. So they're not going to change the equilibrium. And they're going to speed up achieving that equilibrium by lowering that activation energy. Now, enzymatic reactions. So that's kind of the nuts and bolts of like how they're going to speed things up. If you think about an enzyme catalyzed reaction, reactant A going to reactant B. At equilibrium, your concentration of a reactant is going to be equal to the concentration 5 seconds later. Because remember, this reaction is at equilibrium. Now, the concentrations of your product are going to be the same because it's still at equilibrium. At any amount of time after your reaction has reached or equilibrium has been achieved, those concentrations are effectively going to be equal because remember, it's at equilibrium. Now, unless the free energy of the reaction is zero, it's wrong actually to say anything like, well, A is equal to B. The no, the reaction is actually at equilibrium. So if the reaction is at equilibrium, concentrations are effectively never going to change of A because the flow from reactant A to B is a net zero change. Now, that does not mean that our reactants are equal to our concentration of our reactants are equal to one another, though. Now, when it comes to understanding, we'll get more into equilibrium and calculations related to that later on. What I want to take away from today is some of the kind of models that are involved in um, enzyme catalyzed reactions, specifically with respect to substrate binding. A There's a couple of different models of enzyme catalyzed reactions. Well, one of which is that a single substrate can be converted to a single product. An enzyme can grab one substrate and make it a different product. The end. So think about that as like an enzyme that isomerizes. The exact same atoms are present, they're just rearranged a little bit. There are some enzymes that isomerize one molecule to a different isomer. A single substrate can be converted to two products. So a molecule can have a bond broken and two products are released from that. Another reaction is multiple substrates can be brought together to make a single product. Does anyone want to guess what the last combination is? Spoiler, there's one more. Multiple substrates can become a single product. Single substrate can become multiple products. Multiple substrates can form multiple products. You're absolutely right. And we denote that with something like A plus B yields C plus D. Now, this is the one, this last one, that gets kind of interesting. And the reason it gets kind of interesting is because, well, there's a couple of different ways that this can operate. So what I'm going to do here is I'm going to show you guys, again, that Cleland notation. But I'm going to, actually, I can give myself a little bit more space. That's good. That's good. And that's pretty good. So I'm going to show you an ordered reaction, a random, and a ping pong reaction. So an ordered reaction is also known as a ordered sequential reaction. Okay. Now the important players here are E, which is our enzyme. And then I'm going to go with S, or no, I'm going to go with E, A, B. I'm going to say substrate 1, substrate 2. Okay. If I say something is ordered or ordered and sequential, what do you think that means? There's an order and a specific sequence. So whenever I think of like a specific sequence, I think, well, this event has to happen, then this event has to happen, then this event has to happen. I got a private message to clarify. It's these reactions types that are what lower the activation energy once the enzyme binds to the substrate's active site. So these reaction types are all different ways in which an enzyme can take a substrate and make a product. And so an enzyme is, by its kind of nature, an enzyme, yeah, it's lowering its activation energy. But this is This is absolutely relevant to that activation energy changing, but take we're right now taking more of like a 5,000 foot view of substrates being converted to products. Okay. Thank you very much for asking because yeah, thank you for asking questions. Okay. So what I'm going to do up here is give you an ordered sequential reaction. A little bit ago, we looked at a free energy diagram, and we looked at the x-axis of that free energy diagram. That showed us the reaction progression. So from the start to the finish. Does everyone follow that? Okay. So I'm going to say start and end. Okay. Now, for this ordered sequential reaction, what I'm going to do is I'm going to say, all right, well, I have E. That's my enzyme. I'm going to put that on my axis here because my free enzyme is floating around in solution. And let's say hypothetically, well, in an ordered sequential reaction, our enzyme has to bind substrate number one or substrate A. So when our enzyme binds substrate number one or A, the complex that forms is EA. Now, the next thing that's going to happen is substrate B can bind. What is the complex that forms after B binds to E and A? EAB. Perfect. Okay. Now, before you proceed from there, this is an ordered sequential reaction. Could the free enzyme E bind to B alone? You're absolutely right. That is like the heart of an ordered sequential reaction. The enzyme has to bind A in order for B to then bind, because this is an ordered sequential reaction. Now, as soon as EAB, this complex right here, has formed, well, that's when the magic can happen. And let's go with EPQ. Our products are P and Q. I'm just using placeholders there. So this is where I'm going to say chemistry occurs. Many chemists would say that's the respect that you, Joel Gray, have for the field of chemistry. You're just going to say chemistry happens? Yes. As soon as EPQ has formed, P and Q are my two products. Well, these are going to be released in a specific sequence. Let's say Q gets released. Then I have EP. Then P can be released. And then I'm left with E. And what can that E then do? Start all over again. Okay, so in this particular, or in this example, these arrows are indicating this substrate binds, and then this substrate binds, and then this product is released, and then this product is released. If you were to see a model like that, some of the questions you might, or you could consider expecting from me would be something like, um, you will never see the EQ complex formed. True or false? True. You will never see E and Q. You'll see E and P, but Q is going to be the first product that's released. Now, when I've taught this in the past, sometimes people get caught up with, oh, does A become P or Q? Don't get distracted by that. This is just saying one substrate binds, then the next substrate binds. Then one product gets released, and then the next product gets released. I don't want you to get distracted and thrown off by those kind of questions. Does everyone follow me there? The important part about this is that this is an ordered sequential reaction. Specific events have to go in a designated sequence. Now, with that said, I'm going to erase that line because we've got a starting point and then we've got an ending point. Now, this model is the random model. Let's use the exact same combinations of substrates and products. Where it gets complicated, so could you release AB without E? Release A. Well, A and B are my two substrates. So EAB has to form in order for products to be generated. So we'll use the exact same notation that we used in my ordered sequential model for a random model. So I'm going to start out with my free enzyme, E. Is there significance that I wrote the E above the line? No, there's not. Whenever it comes to the free enzyme in a random model, could my free enzyme bind B first? Yep. Could it bind A first? Yep. It's flip of a coin. In which case, we've got two different directions it could go. One direction is A binds, the other direction is B binds. And we will make either EA or EB. Once we have EA or EB, let's say that we have EA. What is my next substrate that's going to bind? You heard it here first, folks. B. Okay, if I have EB, what's going to bind? That's right, your old friend A. In both cases, we're going to get a complex that has both of my substrates and my enzyme. And for the sake of, you could look at that as EAB or EBA. It doesn't really matter one way or the other. So I'm just going to say EAB. My reaction is going to take place. I'm going to get EPQ. Now, this is a totally random output. Either Q is going to be released or P is going to be released. If Q is released, then I have EP. If P is released, then I have EQ. If EQ is produced, then Q is going to be released second. If EP is formed, P is going to be released second. Either way, we end back at the same place. Free enzyme. Ready to rock and or roll again. Okay. Now, can I get a thumbs up or a yes or something that, yes, I'm good, I follow. Okay, great. Now, the last model that I want to show you is the ping pong model. And in the ping pong model, the sequence in which the enzyme and substrate bind varies a little bit. And maybe the most important part about the ping pong model is, I'm going to say the enzyme gets modified. Okay. We'll start and end here. In the ping pong model, we start out with E, our free enzyme, and we bind to A, our first substrate. And in this case, we have EA, but then something happens. Our first of our reactions takes place. So EA becomes, let's say, FP. And to provide a little bit more detail, E becomes F. So the way that I think is helpful to kind of visualize this is a functional group, we'll say. I always like to think of a methyl group or a carboxyl group, goes from A and gets transferred to our enzyme. So something from our substrate gets added to our enzyme. So our enzyme is no longer our enzyme. Instead, it's our enzyme with a phosphate, with a carboxyl, with a methyl group. And when we have this FP, well, our substrate has changed, our enzyme has changed. And at that point in time, we're ready to release our first product. Our first product is P. That gives us a different form of our enzyme known as F. F is uniquely capable of binding to B, our second substrate. So now we have FB. That FB undergoes a change to make E, let's go with Q. And here we've got our second reaction. F becomes E. What's our second product here? Q. Q gets released, leaving behind our free enzyme. So it can undergo this all over again. So these are kind of the processes that enzymes go through in order to catalyze specific reactions. I think that it's helpful to try to fill in some of these gaps with, well, what's actually taking place here? One good way to look at it, I think, is to um, try to process this information and say something for our ping pong reaction with, well, two reactions take place. One reaction in one substrate has to be completely processed in order for my second reaction to take place. Questions that I like to see regarding that ping pong reaction or that I will ask from time to time are the free enzyme E can bind B alone. True or false? That would be false. Only the modified form of our enzyme F is capable of binding B. Okay. So these are ways in which enzymes will bind to substrates and act on them. We're not yet looking at, well, what are the actual electrons that are being pushed here or there, or anything like that quite yet. We're getting there. But for the time being, this is all that we're kind of shuffling around, kind of a, like I said, a 5,000-foot view on this. Does anyone have any questions about substrate binding and types of reactions? So for the exam, would we have a scenario-based question to figure out the type of reaction and steps? There might be something like that. So I'm happy that you asked that. There's a couple of ways that I could ask this sort of question. One way that I might ask a question about these different types, the ordered sequential, random sequential, or the ping pong mechanism, is I would say a modified form of an enzyme is required in order for this type of reaction to occur. And if the enzyme gets modified, a modified form, that would be that F, so the ping pong mechanism. In the third model, does enzyme E come back to its original state or a different one? My drawing wasn't particularly great, but it does return to its original state E. So it can continue this process all over again. Absolutely. Now, what I'd like to do now is I'm going to jump ahead just a little bit. because I've got some examples of our different mechanisms, but what I'd like to do is talk about a couple of graphs real quick. And I just want to introduce you to these and bring up some important characters. So, Michaelis-Menten kinetics. That is the model of enzyme kinetics that most commonly we study. Um, Menten, these are named after two people. I believe they are Canadian, and I think they were at the University of, it's not Guelph, it's in Montreal, McMaster or something. I can't remember. Maud Menten, and I believe Maud Menten's husband um, are the people who this is credited to. And what they studied were a couple of different things. This graph right here illustrates, well, basically a enzyme catalyzed reaction watching a couple of different variables. One variable is your substrate concentration. The next is your enzyme concentration, your free enzyme concentration, then your product and your enzyme substrate concentration. What I want you to recognize about this is initially, if we look at our x-axis here, our substrate concentration is at its absolute highest at the, time point 0 seconds. Our product concentration, product, y-axis is concentration. Our product concentration is at its lowest at that point in time. Our free enzyme concentration is at arguably its highest, but then it dips down kind of inversely as the enzyme substrate complex goes up. Now, our free substrate concentration is going to drop down and it's going to stay down because that substrate is going to be processed and utilized to produce our free product. So these four variables, substrate concentration, product concentration, enzyme, and enzyme substrate concentration, are all important variables to consider. Now, this graph right here is showing concentration of all of these different things versus time. So as the reaction is actually progressing. At some point in time, what's going to happen to all of the substrate? It's going to all be consumed, which is where we're right over here. All of our substrate has been utilized to produce as much product as possible. Now, when it comes to Michaelis-Menten kinetics and studying a Michaelis-Menten curve, I want to bring you on to this right here. This graph right here has a couple of different variables that are important to recognize. The label for our y-axis is initial velocity, or V sub 0. The label for our x-axis is substrate concentration. Now, there's a couple of things to keep in mind with this. All trials have fixed enzyme concentration. Okay. Then what I think is when you hear initial velocity, or personally, whenever I hear of initial velocity, I think of someone doing a sprint, or I think of sprinters. When are they presumably at their fastest? When they're releasing from the starting blocks. In the event that you're a sprinter and you're thinking, no, you're not right, you're absolutely right. I'm probably not right. But I'm thinking about this conceptually. I have the most energy when I begin my sprint, not half a second into it, not a minute into it, at the very instant that it kicks off. That's when I have my burst of energy and then it craters. So our initial velocity is when we're at our highest. Now, in order to study an enzyme, let's go ahead and set up a series of trials. T1, T2, T3, T4. When I was a graduate student, I had a professor who would give this example, and he would use Oreos because I like Oreos. And the enzyme that we used was an enzyme called Oreo cookie lyase. And what Oreo cookie lyase does is it takes an Oreo and twists to split them. You twist an Oreo to split it. So we could say that our substrate is our, let's go, our substrate. Okay. Our substrate is our Oreos, our cookies. The enzyme that we would use are the people who twist Oreos. So you twist them open, and boom, where you get your product. So the enzyme are Oreo twisters. I'll say Oreo twisting people. Then last but not least is our product. What's our product? The split Oreos. Okay, so our substrate was the Oreo, the person is the enzyme, and the product is the split Oreos. Okay. So in every trial, we have a fixed enzyme or a fixed concentration of enzyme. Let's just say that our enzyme is 3 people. That's not going to change, right? Then our substrate. Well, our substrate is going to be the number of Oreos that you give to this enzyme, and then we can change that. So let's say we've got 3 people, and with those 3 people, we're going to give them a time. We're going to set a timer for 10 seconds. And among those 3 people, we'll put down 1 Oreo and say, okay, you got 10 seconds, twist. Okay, so let's say that's 1 Oreo. Among those 3 total people, have you limited your substrate? Yeah. What's our product going to be after 10 seconds? We're going to be able to split 1 Oreo. Now let's say that we put 3 Oreos down. We got 3 people, 3 Oreos, give them 10 seconds. What's going to happen? We're probably going to get 3 Oreos. Let's say that we put, for trial number 3, we put 9 Oreos down. Okay, we got 10 seconds. Let's say that they can get through 7. Maybe there's some infighting and talking and everything like that. Because, you got the 9 Oreos, grab, twist, grab, twist, grab, twist. 3 people, yeah, let's say they get 7 done. We increase our Oreos. We'll go up to trial number 5. Let's go to 12, and then let's go to 24 Oreos. When you put 12 Oreos down, maybe we'll get 9 done. Is everyone willing to grant me those numbers? Okay. If we push it all the way up to 24 Oreos, are we going to get 24 products? Nope. Chances are we're going to kind of plateau in our product generation. So I think that it's pretty reasonable to say that we'll get about 10. And if we continue increasing this number of Oreos, what's going to happen to the number of products? Well, the margin is going to, the increase is going to be very marginal. And so you're going to see data, so our initial velocity versus our substrate concentration. The velocity would be the number of Oreos done in 10 seconds. We're going to see that velocity, the number of Oreos increase. But then at some point, what's going to happen to the rate of production? It's going to plateau just like this. You could give trial number 5, 3 people, let's give them 10 million Oreos. you're still going to get about 10 Oreos done. At some point, it's got kind of a diminished marginal return. You're just going to kind of plateau. Okay, that's where I want to stop because it is 7:50. I'm sorry for stealing extra of your time. Um, but I am going to stop my recording right there, and we will pick up from there on Wednesday.